Today we are going to learn about the packaging of DNA. How DNA is packed inside the nucleus, because we all know that DNA are on chromosomes and chromosomes are present in the nucleus. So we will see how this DNA is present in the nucleus, how this DNA is packaged. Now, why it is a concern of packaging of DNA is there, because the length of the DNA double helix in a cell can be calculated by multiplying total number of base pairs with distance between two consecutive base pairs. Now we know that the distance between two consecutive base pair is 0.34 into 10 raised to the power minus 9. And total number of base pairs also we know for different organisms, and so we can calculate the length of the DNA. So this is the formula to calculate the length of the DNA: total number of base pairs into distance between two consecutive base pair, that is 0.34 into 10 raised to the power minus 9. This we have done in our last class when we discussed about the salient features of DNA. Now the pitch, which is having 10 base pairs, and the length of the pitch is 3.4 nanometers. So the length of DNA in human cell is 6.6 into 10 raised to the power 9 base pair. Now this we have come to know from the length of the number of base pairs. We have come to know that haploid human cell is having 3.3. So the diploid, because the human cell is diploid, so it would be 6.6 into 10 raised to the power 9 base pairs. So these are the number of the base pairs. Now when we multiply this number of the base pair with the length, the distance between two consecutive base pairs, that is 0.34 into 10 raised to the power minus 9 meters per base pair. So it will come to 2.244 meters. That means it is more than 2 meters. And the dimension of a typical nucleus is approximately 10 raised to the power minus 6 meters. So how is it possible that such a long strand of DNA is present in the nucleus? So that is a big puzzle to be solved. So the question arises that how such a long polymer is packaged in nucleus? How can more than 2.2 meters is getting fixed into a nucleus which is having a dimension of 10 raised to the power minus 6 meters? So this is the big question. Now we will see that how DNA is present in prokaryotes and in eukaryotes. Prokaryotes, you know that prokaryotes are the primitive cells which do not have any well-defined nucleus, and they do not have membrane-bound organelles. So these prokaryotes, they do not have a well-defined nucleus, but still their DNA is not scattered throughout the cell. Their DNA is not present all over the cell. Their DNA is present in the center only. And how it is present? The DNA is a negatively charged molecule. You know, phosphate is present, and that is negatively charged. So overall DNA is a negatively charged molecule, and being negatively charged, it is held with some proteins. Proteins are positively charged. So the proteins which have positive charge on them, they are together with this DNA which is negatively charged. So this DNA which is negatively charged is held with some proteins that have positive charge on them. And the region where DNA are present with proteins is termed as nucleoid. So the region where DNA is present in a prokaryotic cell is called nucleoid. Here, so we are talking about prokaryotic cell. Here the DNA is not scattered, though there is not a well-defined nucleus in prokaryotic cell, but still the DNA is not scattered. The DNA is present, held by the protein, the positively charged proteins, and the center, and this is called nucleoid. So the DNA in nucleoid is organized in large loops held by proteins. So most prokaryotes, they carry a small amount of genetic material in the form of a single molecule or chromosome of circular DNA. The DNA in prokaryotes is contained in the central area of the cell, which is called the nucleoid, and which is not surrounded by a nuclear membrane, because this is a prokaryotic cell. So have a look on this diagram. This is bacteria, and this is a prokaryotic cell. So in this prokaryotic cell, this is the nucleoid. The nucleoid is present here, and this nucleoid is DNA with protein. DNA is negatively charged and proteins are positively charged. See here also, this is again a bacteria. The nucleoid is present here. This is DNA and this is called nucleoid. Now, in case of bacteria, you will find this is called chromosomal DNA, and there is a circular DNA also, which is present here, and this is called plasmid. So, circular DNA is also present, and this is chromosomal DNA, which is present with the positively charged proteins, and this is called nucleoid. Now we will see how DNA is present in the eukaryotes. In prokaryotes, it is rather simple, but in eukaryotes, this is much more complex. The organization of DNA in eukaryotes is much more complex, because in eukaryotes, you will find a well-defined nucleus, and inside that, this DNA is present. So in eukaryotes, there exist positively charged basic proteins, and these proteins are called histones. Now DNA wrap around the histone octamer. Octamer means octa is for 8 and mer is for unit. So 8 units of protein together, 8 histone proteins together is called histone octamer. And this histone octamer, the DNA is wrapped around this, like you are wrapping some thread around a solid thing. So similarly, this is the octamer, and DNA is wrapped around this octamer, and this structure which is formed is called nucleosome. Each nucleosome is containing 200 base pairs of DNA helix. Now, nucleosome in chromatin are seen as beads on string under electron microscope. See, if you compare the DNA in prokaryotes versus eukaryotes, in prokaryotes you will find nucleoid, whereas in eukaryotes you will find chromatin inside the nucleus, and this chromatin is present as nucleosomes. Nucleosomes are like beads on a thread, and this whole thread like structure condensed is known as chromatin. So this is the difference between the packaging of DNA in prokaryotes and eukaryotes. Now, histones are rich in the basic amino acid residues. As you know, that proteins are made up of amino acids, because proteins are the units for proteins are amino acids. So histones are the amino acids which are basic amino, rich in the basic amino acid residues, lysines and arginines. So lysines and arginines are the basic amino acids which are present in the histones. The histones are made up of these. Now, both the amino acid residue, they carry positive charge. What are these both the residues? Lysines and arginine. They both carry positive charge in their side chains. So they are positively charged. DNA is negatively charged. So they are together. They attract each other and they are held together. So these histones are organized to form a unit of 8 molecules. 8 molecules of histones, they come together and form a unit. And this unit is called histone octamer. This unit is called histone octamer. Octa is 4. Now, negatively charged DNA wrapped around the positively charged histone octamer, and this is forming a structure which is called nucleosome, which is called nucleosome. In case of prokaryotes, it was nucleoid, and that was protein with the DNA. Here, the proteins are histones, and they are present in a octamer unit, which is called histone octamer. And this histone octamer, around that the DNA is wrapped, and this structure is called nucleosome. Now see, it looks like this. Can this just count this 1, 2, 3, 4. Similarly, 4 below. So 8 histone proteins. So this is called octamer. Octamer, and this thread-like structure which you can see, this is the DNA. This is DNA. And this is again another protein which is not included in the nucleosome. So nucleosome is 8 histone and these 2 wraps of double wrapping of DNA around it. This is your NCERT diagram. This is H1 histone. This is, they have given us single circular structure. This is showing the octamer, histone octamer. So this is actually not a single unit. It is consisting of 8 histone proteins as a unit, and this is called octamer. So this complete structure is the histone octamer, and there are this is the DNA doubly wrapped around this octamer, and this is the what we can observe under the electron microscope. These beads like structure, these are nucleosomes, and these nucleosomes on thread like. So this is EM picture. EM is electron microscope. So under the electron microscope, you can observe chromatin like this. You can see these beads like structures. And what are these beads? These beads are this nucleosome. So nucleosomes are seen in under the electron microscope like that. Now, what is the first level of packing? Packing is nucleosome. What is nucleosome consisting of? Nucleosome is consisting of DNA and core histones. Now, what are the core histones? Core histones are H2A, H2B, H3, and H4. So, all 4, 2 each. So, total 8. 2 H2A, 2 H2B, 2 H3, and 2 H4. So, these 4 histones, they are forming the octamer, histone octamer. So, DNA wrapped twice. In all diagrams you have seen twice. So, twice the DNA is wrapped. 1 and 2. Twice it is wrapped around the octamer of core histones, and these core histones are consisting of this. And the structure, the yellow structure which you can see, this is not a part of the nucleosome. It is attached to the DNA near the nucleosome. So this is not the part of the, this is a histone protein only, but this is not the part of the nucleosome. Nucleosome is consisting of 8 histones, and this octamer and the DNA is wrapped around it twice. Now see this. This is again this is the twice wrapped DNA, and these are octamers, histone octamers. So this histone octamers, if they are very closely associated to each other, then it is closed chromatin, and if they are slightly away from each other, it is open chromatin. This is another nucleosome. This is nucleosome, and you can find the 8, 1, 2, 3, 4, 5, 6, 7, 8. 8 histone proteins, and this is surrounded, wrapped by a DNA. So this structure is called nucleosome. Just to make it more clear, 1, 2, 3, 4. 2 of H2B, 2 of H2A, 2 of H4, 2 of H3. 8. 8 histone together, and this DNA twice wrapped twice. This is nucleosome. Now see, in prokaryotic cell, this is the chromosome associated with the proteins held by the protein. In eukaryotic, you will find histones, and these histones, they are like this. So these are the small structures, octamers, and they are held closely with each other, and they are forming the chromatin. Now, typical nucleosome contains 200 base pairs. If you see one nucleosome, it contains 200 base pair of DNA helix. Now, repetitive units of nucleosome. Nucleosome after nucleosome, nucleosome after nucleosome, repetitive units of nucleosome, it forms the chromatin in nucleus. That is called chromatin. And this chromatin are thread-like stained bodies seen in the nucleus. So under the electron microscope, we are seeing this, the thread which we are seeing is chromatin, and the beads which can see are nucleosomes. So threads on beads on string, that is what is the structure which is seen under the electron microscope. So this chromatin are thread-like stained bodies seen in the nucleus. Now nucleosomes in chromatin are seen as beads on string structure when viewed under electron microscope. And chromatin fibers are further coiled and condensed at metaphase stage. If you remember, when we were doing cell division, in cell division prophase stage, we say that the genetic material start condensing. So this is the genetic material which starts condensing at prophase, and at metaphase it is further coiled and to form the chromosomes. So at metaphase stage, you can observe chromosomes. So this chromatin fibers, they are further coiled and condensed at metaphase stage of cell division, and they form chromosomes. The packaging of chromatin at higher level requires, we saw this is the primitive level, the primary level of packaging, when the histone octamer is there and the DNA is wrapped twice. The packaging at higher level requires additional set of proteins, and this additional set of proteins are collectively called as non-histone chromosomal proteins. They are non-histone, so this additional set of proteins which are required for packaging at higher level, they are called non-histone chromosomal proteins, and they can also be called, the abbreviation is NHC. So NHC proteins. Now this chromatin, if you see, can be divided into two depending upon how they look and how they stain. So euchromatin and heterochromatin. Euchromatin is the loosely packed regions of chromatin, which stains light. That means the chromatin which is taking very less stain is euchromatin. And this euchromatin is transcriptionally active. Active means this is going to transcribe into messenger RNA. So transcriptionally, euchromatin is the active chromatin. And if you see the heterochromatin, heterochromatin is densely packed region of chromatin, which is the chromatin is densely packed and it stains dark. If you see it under the microscope, under the electron microscope, you will see the dark stained bodies, and this is heterochromatin because they are densely packed, and this chromatin is transcriptionally inactive. That means they are not going to transcribe into messenger RNA, whereas the euchromatin is the chromatin which is transcriptionally active chromatin. Now, this is the summary of packaging of DNA helix. DNA of eukaryotes is wrapped around positively charged histone proteins to form nucleosome. Now, nucleosome contains one nucleosome, contains 200 base pairs of DNA helix. So you can calculate how many nucleosomes can be there in a nucleus because we know the number of base pairs. Histone octaber is what? H2A, H2B, H3 and H4, all 2. So total 8. Now linker DNA bears H1 protein. H1 protein is present, but that is not part of the nucleosome. Now chromatin fibers formed by repeated units of nucleosomes. Non-histone proteins are also required for packaging. When the higher level of packaging is done, non-histone proteins are also required. Regions of chromatin loosely packed and stains lightly are called euchromatin, and regions of chromatin densely packed and stains darkly are called heterochromatin. So the chromatin can be divided into euchromatin and heterochromatin as per the stain absorbed and whether they are transcriptionally active or inactive. So this is how DNA is packaged inside the nucleus.